I'm Regan, and I'm going to talk to you today about the extent to which local adaptation is associated with range expansion. And so if you can imagine, this is a species range, its geographic distribution in space. For many species, these range edges remain stable over very long time periods, but under climate change, some species ranges are actually expected to shift in space. And so, for instance, as it gets warmer further north, species ranges may contract from the bottom, where it's getting too hot and dry. And so they would have to track the warming climate and shift their ranges northward in order to prevent them from collapsing in on themselves. But it's unclear the extent to which local adaptation will be involved in species expanding or shifting their ranges. And this could actually be important, especially for longer-lived species who might take decades to adapt to local conditions. They might not be able to do that fast enough to keep pace with the rapidly changing climate. And there is evidence that adaptation has been required for range expansion in some systems, particularly invasive species. So for instance, purple loosestrife, which is shown here, Claudia and Barrett did some work that showed that it was locally adapting quite quickly as it spread throughout its invasive range in North America. And there was also a meta-analysis here by Oduar et al. That showed they compared native and invasive species, and they found that invasive species were just as locally adaptive as native species. And so clearly they've had less time to adapt, but they were just as locally adapted as the native species. So clearly local adaptation can occur quite quickly to allow colonization and range expansion, which would allow for the necessary range shifts to track the rapidly changing climate. And it makes sense that adaptation might need to occur in these cases because just because it's getting warmer further north doesn't mean that the entire habitat is being picked up and moved further north. So there could be other conditions that the species would need to adapt to. And so I wanted to examine this using a plant that my lab has worked on for a while, the Pacific coastal dune endemic Camisoniopsis strathifolia. It's pictured here, and it grows along the west coast of North America, so from about mid-Oregon down into Baja California. It's got a fairly linear range along the dunes of the coast, and the northern range limit has been stable for about 150 years, according to herbarium records. It's a short-lived perennial, so it usually does most of its reproduction in its first year, but it can live up to 3 years. And it grows in this kind of patchy dune habitat. It'll grow mostly in the open sand patches shown here. And so a previous student in our lab, Karen Samus, did a beyond range transplant experiment using this species in 2005. She was looking at the mechanisms that limited the species' northern range limit. And so she took individuals from these 8 source populations shown here on the left, and these color codes and site codes will be consistent throughout the presentation. So we're moving from warmer red colors in the south up into cooler blue colors in the north. And the site codes, W just means within the range, and then the number is the distance in kilometers from the range limit. And so this is the range edge, E for edge, 0 kilometers from the limit. And so Karen took individuals from all 8 of these populations and she transplanted them back into 5 sites shown here on the right. And so we've got 4 within range sites, and then we've got the beyond range site as well. So it's beyond the range 60 kilometers. And she monitored the individuals for the 3 years following planting to get their lifetime fitness. And interestingly, she actually found that fitness increased toward and beyond the limit. So particularly for individuals from the 5 northernmost populations here, she found that fitness was actually higher toward and beyond the limit. The highest fitness was found at the beyond range site. And then for the 3 southernmost source populations, fitness was not significantly different below or beyond the range. And notably also, there was no local adaptation found within the range. So when she took individuals from the within range sites and then transplanted them back into their home site, she didn't find that they were outperforming the individuals from the foreign populations. And so as well, we went back during the last 3 growing seasons, and we found that actually fitness and abundance have remained high beyond the range after about 12 generations have passed. So they've been persisting there with high fitness for quite a long time now. But what we really wanted to know is whether the beyond range population actually had to adapt in order to persist there over multiple generations. So if you recall, there wasn't much signal of local adaptation within the range, but we did see those northern populations outperforming the southern ones at the beyond range site. And we do have reason to suspect that local adaptation might occur in this system because Karen looked at the plant community composition throughout the northern range of the species, and she found a lot of ecological variation. So the plant community changed a lot across the northern range and across the range limit of the species. And you can kind of see that here. This is the southernmost source population, and you can see that the plant community and the dune system differ a lot from this is the beyond range site pictured here. So there is that ecological variation, and then there is also genetic variation for fitness among individuals. Like I said, the northern individuals were outperforming the southern ones. And so we've got both that ecological and genetic variation, so both of the requirements for local adaptation are met. And additionally, since we mixed so many genotypes together at that beyond range site, that would also increase the genetic variation and possibly further facilitate local adaptation. And so I wanted to investigate this using another transplant experiment. So I did this in 2018. I took individuals from all of Karen's original source and transplant populations. So the circle points here are locations where I was just able to take from the pure natural source populations. And then the triangles, I was able to sample from both the natural population and the mixed genotype transplant population. And then I also took individuals from the beyond range site. And then I transplanted them all back into 5 sites. So here we've got 3 within the range, and then the original beyond range site, and then an additional site about 200 kilometers beyond the northern limit. And I planted a total of about 2,800 individuals across all of these sites. And so I'm going to show you some very preliminary results from this. The plants are still surviving out in the field. This will be their last growing season, so I'll get their final fitness in November. So this is just the first 2 years of fitness. And so I first just wanted to look at the data as a sort of repeat of Karen's experiment. So I've got one less within range site here, and then I've got an additional beyond range site, but I just wanted to see whether I found that same pattern of fitness increasing towards and beyond the range limit. And so on the y-axis here, we're going to have the mean reproductive output. So for each site source combination, there's going to be a point that represents the mean reproductive output for those individuals. And all of these source populations are just going to be the natural populations. So you're not seeing any data from the individuals that I took from the mixed genotype transplant populations. And this data does include zeros for plants that died before reproducing. So it does include that element of survival. And so here I've just put on one source population to start. So this is the individuals taken from the edge site, the range edge. And you can see at these 2 within range sites, when we plant individuals there, the fitness is pretty low, and then it increases towards the range limit as well as beyond the range. And then we find that at the far beyond site, it actually declines a bit. So that's one site, and then I'm just going to throw all the rest of the natural populations on here. And it's a lot of data, a lot more noise, but you do still sort of pick up that trend. So the fitness seems to increase toward and beyond the limit, especially at that original beyond range site, it increases or remains high. But then at the far beyond site, we do see a bit of a decline in fitness there. And so we have found kind of a repeat of Karen's results where fitness increased toward and beyond the range as well. And notably, again, I wanted to see whether those within range sites had shown any local adaptation. So if you recall in Karen's, there was no local adaptation. And here we find that again. So the green point here is individuals taken from W133. And we see that when we plant them back at their home site, they aren't outperforming the rest of the individuals. So they don't have higher reproductive output than the other foreign populations. And same at W36, we've got the purple point not outperforming the rest. And then at the edge site, we've got the dark blue point. And again, it's not outperforming the rest of the individuals. So again, we're not seeing a signal of local adaptation here in the natural within range populations. So let's see what happened at that beyond range site. And so for that, I'm just going to be using the data from the mixed genotype transplant populations. And that's important because this way I'm controlling for the increased genetic variation from mixing genotypes and just comparing that beyond range transplant site to individuals from the within range transplant sites. And so that's what we've got here. Each point is the mean from a different source population, and the source populations are Karen's original transplant populations. So all of these have many populations of mixed genotypes in them. And the black point here is those individuals taken from the beyond range site. And what you will notice is that at the Beyond Range site, when we plant individuals from the Beyond Range site, we do see that they are outperforming the rest of the individuals. So they've got higher reproduction than the other 4 transplant sites. It's not significantly different from these 2 higher ones, but it is the highest, and so there is maybe that small signal of local adaptation to the Beyond Range conditions, which I thought was really cool, especially when we compare it to the within range sites. So again, all of these populations had that mixing of genotypes, that increased genetic variation, so they all had that potential to locally adapt, but at the within range sites, we don't see it. So the green point again at W133 is just in here within the mix of the others. It's not outperforming. Same with purple at W36, and same here with the dark blue at the edge site. It's not outperforming the rest. So it's really the only place that we're getting that signal of local adaptation is at the Beyond Range site. But again, I do have to wait for the final lifetime fitness this November and do a more intricate analysis to determine for sure. And so just to sum up, first of all, I found evidence that supported the original conclusions from Karen's transplant experiment that fitness actually increased toward and beyond the limit. And it's really rare to have these big transplant studies repeated in multiple years. So I thought it was really exciting to be able to repeat Karen's results. And secondly, I found some very preliminary evidence that the population actually may have adapted to those beyond range conditions in order to persist there over all those generations, which I thought was kind of remarkable and really shows that even in species that aren't locally adapted at all within their ranges, they may still need to adapt in order to expand their ranges under climate change. And so with that, I'd just like to thank my field teams, Karen Samus, who did all of the original transplant work, these governing bodies for permits, NSERC for funding, and thank you all for listening, and feel free to send me any questions. Thank you.